Thanks for the invitation. Thanks to you and Jeremy for the invitation. And it's particularly nice to come to this meeting because, yeah, as Sammy just mentioned, the project I'm going to talk about was to a significant extent funded by the network. So it allowed us to do something which it would really have been quite difficult to do otherwise. And in essence, what we wanted to do here was to see how good predictive models are at the moment. So if you have a data set and you ask a research question, and anybody can participate in answering that research question, how good are the results? And we wanted to do that by asking people to predict molecules and then making molecules and evaluating them. So we sort of close that loop. And so I'll talk a little bit about that today. The area I'm going to be talking about is drug discovery, small molecule drug discovery. I'm an organic chemist by training, so any technical knowledge of the methods that were employed to answer this research question, I may not be terribly hot on. And I guess I'm speaking from the user perspective here. So as an active researcher trying to discover small molecules to cure disease, what can I call upon? How do I collaborate with people with expertise in machine learning or AI that I don't have? So hopefully it'll be an interesting case study. The project took part in something called Open Source Malaria. So for those of you who are not familiar with Open Source Malaria, there was a question just earlier about open source research. This has been a drug discovery project which has been ongoing for a few years now, and which over the years has had contributors from high school students through to senior professionals in the pharma industry. A few hundred people have contributed over the years to this. And we run with these 6 laws that are shown there, and which still nicely encapsulate how everything works. So all data are open and all ideas are shared. Anyone can take part at any level, and there are no patents, just to make it clear to people that this is the way it's going to work. And the other laws there are about how to behave as part of the consortium, really. The 6th law is important about ownership, that the project stands on its own and is not owned, or at least too heavily owned, by any one person in particular. So it's a similar structure to Wikipedia, really, where anyone can contribute. And the point is that the research takes place sort of live and online. So if you want to participate, you can, and you don't have to wait for it to finish before you do that. So it is very, very much a sort of participatory sport, as it were. What that means is we need open lab notebooks. So the lab notebooks that are used by participants, including all the students, all the participants, are online, so you can see those, and they can be searched by Google, so you can find things quite easily. The discussion that we engage in is crucial, and in the early days we really struggled with this, about how to have a good, solid discussion online with pictures and links and everything. But we've discovered that the discussion forum, the issue tracker on GitHub, which many of you will be familiar with, is really good as a way of figuring out who's doing what and when and what's left to do. Open data is very simple for us in that we just use a Google Sheet to paste all the data into, including all the smiles and inchy strings and so on that make it more machine readable. And then for a community, we use social media, which has transformed our ability to kind of reach out to people. And so what's happened over the years is with this project and with related projects, what happens is people spontaneously give. And again, anyone who's got any familiarity with open source software will not be surprised by this. People spontaneously give if they know that their contributions are required and worthwhile. And they know that the project is live and online. So there are no outsiders, if you will. And so over the years, we've done a few different series for small molecule antiplasmodium compound discovery. The first 3 series we looked at were derived from compounds that were put in the public domain by GlaxoSmithKline, and they did a big screen of their compound library against the parasite. These, as will become important later, these were phenotypic. This is phenotypic drug discovery. In other words, you have a compound that works, has a desirable biological effect, in this case, killing the parasite, but you don't know the target, the molecular target. So your ability to improve the molecules is reliant on a kind of intuition built up over years by the medicinal chemists who are part of the project. So it's not rational drug design in the sense of target-based. fitting something to a biological structure. The fourth series came out of another screen by Pfizer and was put into the public domain. And we've been pursuing series 3 and 4 quite a lot recently. Series 4 is very notable because the molecules are active in an in vivo model, a mouse model of malaria. And series 3 is interesting because it's a fantastic compound and we don't know yet what the target, the molecular target is. Series 4 is the one I'll be talking about today mostly. And it's interesting because there is a suspected target for which we don't have a structure. So just to give you one example of how this works generally, this is an example from the first series from a few years ago, but nicely sort of captures what we do. We needed these 3 molecules to answer certain questions as part of the exploring the SAR of the series. Couldn't make them, so put out a request, to everybody on Twitter for anyone who wants to make these molecules. And someone called Patrick Thompson, who was finishing his PhD, unrelated to what we were doing in Edinburgh, said that, yeah, he can make those molecules. He decided to take one, the sulfonamide in the top right, and make that, and just started working on it independently of us, posting raw data, obviously, so that we could see the quality of the work. And then when he finished, he posted the molecules off to get evaluated versus controls that we were using, and discovered nice, useful data points in the SAR. In this case, inactive data points, but still important. He was trying to replace this troublesome ester on the screen with something that might be better able to withstand in vivo hydrolysis. And so got his name on the paper and was a key contributor. And then years after we'd finished our collaboration, we met and had a beer. But it's this kind of structure allows people to operate independently because they know that what they're doing is required and there's going to be no duplication of resources. So publishing work like this is very important, even though everything is live and in public domain. Publication still remains very important as a sort of drawing a line under when a project has reached a certain milestone. And so the first series we published was published in this paper here a few years ago now, and it's interesting in how difficult it was to publish this paper because there are lots of different contributing data sets everywhere. We even had a conversation on Derek Lowe's blog in the pipeline, which influenced the project. And so we had to sort of screenshot Derek Lowe's blog as a supporting information item. And all the lab notebooks were zipped up and put on a library repository and so on and so on. So it was a lot of work to publish it, but perfectly okay to publish work like this, even though every constituent data point is in the public domain. And in more recent years, I guess it's become more common to have papers that have most of which has been in the public domain, or at least we live in a preprint era where people are a bit more happy with the idea of papers being published in journals, even though they've appeared elsewhere. And we recently published this one on the antimalarial pipeline, where in addition to the article, the paper itself is hosted on a repository, and so it can be easily updated. That's the idea. So as new things are discovered about the pipeline or as drugs move through, we should be easily able to modify the paper and sort of publish an updated version of it. Okay, so just in terms of the specific project we were doing with that I want to talk about today, we were dealing with a series in Open Source Malaria, series 4, which had a great deal of promise. So as I mentioned, we knew that the molecules, 2 molecules in this series, which had been active in vitro, so against the blood stage potency assay for malaria, had shown potency, a great promise in vivo in the mouse model. And we were at that lead optimization stage of a medicinal chemistry project where you're doing kind of single atom changes to try and improve things. In our case, try and improve solubility a little bit, try and maintain potency, and try and reduce metabolic clearance rates. And so some typical examples are shown here where we were messing around with a certain part of the molecule that we knew was important in terms of potency, and we were just trying to engineer in less clearance. So on the molecule on the left there, you can see we're messing around with the benzylic position, which is a known metabolic hotspot, I guess, installing hydroxyls that would help with solubility and so on. In the third molecule there, you can see we try to replace the phenyl ring, which can sometimes lead to less good solubility parameters, I guess, with something that is more so-called sp3 rich, so something which is more hydrocarbon rich, which is maybe more soluble than something with a benzene ring. So these are typical strategies you use in a program like this, where you don't really know, well, you don't know what the molecular target is. So you can't make changes in that kind of rational way. You have to be guided by this sort of medchem intuition that people, particularly in the private sector, in the pharmaceutical industry, are supremely good at looking at a series of molecules without the molecular structure of the biological target and thinking, okay, well, what do we do here to improve things? So we were at that stage. And this is where we are in the series, is trying to just decide on the latest lead compound that fulfills these criteria so we could reevaluate one of these molecules in a mouse model again. That's kind of where we are, and the paper is being written online at the moment. But as you can see from the sort of exploration that we were doing, you have to make a lot of molecules to figure out what's going on. In many cases, even though we had a lot of active molecules, we were still spending quite a lot of time on making inactives. So it would still be the case that the medchem team, including me and the postdocs and contributing people and advisors online, were still suggesting and making molecules that turned out to be surprisingly inactive. And so if you count that up, it becomes quite an expensive endeavor making these sort of inactives. I mean, it comes out to about $2,000 a week or something in terms of postdoc time and everything else to throw at these kind of inactives, which weren't really helping us very much. And so naturally, we wanted to be better at predicting actives, right? Because that's really what we're interested in. We want to be better at doing that, but we didn't have the biological target to guide us. So over the years, we've wanted to be better at this. And so we thought, well, how can we get better at deciding about which active molecules to make and sort of improving the potency without accidentally making inactives? So it's a big question in drug discovery, of course. And it's very common that you have a series of molecules. You don't necessarily know what the target is, and you want to avoid unnecessarily making inactives. So this particular target is interesting. So there is a strong suspicion that we know what the protein is that is being targeted. And it's an ion pump, a membrane-bound ion pump called PF-ATP4. The expert in this area is a guy called Kieran Kirk, who works at the Australian National University in Canberra in Australia. And it was suspected that this was the case because Kieran had an assay which measures ion gradients across just outside the membrane of the parasite. Here it is in a schematic just inside a red blood cell. And it was known that if you give certain molecules, that you block this ion pump, and you can see the changes in the ions close to the membrane. And it was noted in this series that activity in that assay, so disruption to sodium ions and protons, was correlated tightly with killing ability for the whole parasite. So it appeared to be that this was an important mechanism of action, and it appeared to be that this was our mechanism of action. The protein itself, PF-ATP4, is horrifically complicated and membrane-bound. So it's the kind of worst-case scenario for trying to do structure-based discovery. And even though people have tried to derive the structure through crystallography and so on, there had been no success, and still has not been success to date. So we thought, well, this is probably the target. So we're not completely blind, but we still don't have a structure of it. So we still don't have the ability to design molecules rationally bit by bit. And what was particularly interesting about this target is how many different chemotypes appeared to be hitting it. So here are some of them. Our molecule is up on the top left there. Others that are quite well advanced are in the top line. In particular, the second molecule here, this spiroindolone compound, is the sort of next great hope of antimalarial chemotherapy. It's a molecule which is in phase 2B, as I understand it. And it would, if it was cleared and got through to market, would be the next new antimalarial to be approved, and would be the first in many, many years to have a new mechanism of action. So it's quite exciting. But in a screen that Kieran did of a bunch of molecules that were put in the public domain by the Medicines for Malaria venture, many, many different chemotypes, some of them are shown here, also were found to do the same thing, apparently. And you don't have to be a drug discovery person or an organic chemist to see that there is no real structural similarity between any of these molecules. And it was a bit of a puzzle. How could so many structurally distinct molecules be doing the same thing? It's possible to grow resistant parasite lines in the lab through gradual exposure to low levels of particular molecules. And this was done in a really nice paper by Kip Guy and colleagues that came out quite early on. And you can map, this is a homology model that's shown here on the screen of the protein. So it's not a crystal structure, it's a homology model. And you can map where the mutations are that arise when you develop a resistant parasite. And they don't really cluster very well. So lots and lots of different places being altered in this protein appeared to give rise to parasites that were tolerant to these molecules, which was quite a surprise. So no real guidance there. It wasn't as if it was the same site each time, lots of different sites. So we were wondering, well, maybe this is just a very large target and there's lots of internal surface area. And maybe there's lots of different ways in which molecules can bind and somehow mess up this target. We just weren't sure. We had no idea. So we decided to try to do something about this. And early on, we did something very, very simple. So the postdoc who was working on the project, Murray Robertson, did a nice bit of work just with a simple sort of pharmacophile generation protocol using some commercially available software and tried to map, all that was known about what was in this series about what was active and what was inactive, using some randomly chosen molecules from a supplier catalog to as presumed inactives. And so did the usual thing that many people maybe on this webinar are familiar with about developing some kind of pharmacophile model, which is a standard protocol, and tried to be predictive about molecules. But it was a first pass that he did very early on in the project, and was not predictive. So you can see by these structures here, which were then subsequently evaluated, that the predictions are shown. In green, it's a prediction, yes, 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 up here. And when it was measured, the measurements did not align well with the predictions. And one of the reasons we think about this is that the model didn't take into account the possibility of overlapping binding sites, that maybe things are binding in a slightly different place in the protein. And it didn't account for chirality. So the molecules, we know that there are cases where the different enantiomers of a chiral molecule will behave differently, and the model didn't account for that. So there were lots of reasons why this could be improved, but we didn't push this further for a little bit of time because we were busy with other things. But we eventually came back to this and thought, well, maybe we should just frame this as a competition. In many open source projects, you don't necessarily want to dangle rewards in front of people because it changes the reasons why people participate. But competitions can be amazing in terms of what they can do for open projects if you frame the question right and if the data set's good. There are some beautiful examples in the literature over the years. I don't know if any of you are familiar with the maths work MATLAB contests that have been done over the years. Some really wonderful examples where simple algorithmic problems have been set. So the derivation of a simple algorithm to achieve something that's quite straightforward. In some cases, it was about taking a big bank of songs. How do you best fill a CD so that it's maximally used? Or how do you program, this was the one that's on screen here, is how do you program the Mars rover to best explore a big area of Mars and all those things. And the interesting thing about these competitions is that when you submitted a model, everyone else could see it. And anyone else could take your model and slightly modify it and then resubmit that solution as their own. So it was a really neat way of sort of motivating people to do things by allowing them to see what else was going on, which is really neat. So this generated a frenzy of activity. In each of these cases, this competition ran over a few days, and the solutions were kind of iteratively improved until you get to a solution at the end where all the participants were agreeing that they wouldn't have come up with it themselves. They had to sort of see what each other was doing. Some people might say this is parasiting off other people's work. Other people say this is a great example of open science, which is my view. Other examples have been happening over the years. This is a really nice one from Jörg Benzin, who worked then at Boehringer Ingelheim, where a competition was run to try and make sense of a particular physicochemical parameter, where the nature of the parameter was hidden from the participants. So people just had the data and had to try and develop a predictive model for the data without necessarily knowing what it means, and work really nicely using a platform called Kaggle, which many of you may be familiar with. And then just, of course, the other day, we saw the results of the CASP competition of predicting the structures of proteins based on the sequence of amino acids. And the CASP competition has been a fantastically exciting spectator sport for many of us, and is now apparently being solved, in inverted commas, by the DeepMind submission that came through just the other day. Really very obviously commercially important, academically very important result to try and predict well the three-dimensional structure of proteins. But as Al Edwards from the SEC reminded us the other day, the competition is only as good as the data it's built on. And in this case, the CASP competition, of course, is built on an absolute treasure trove of open data, which is, of course, all the structures of proteins that have been submitted experimentally, submitted to PDB and elsewhere. So a nice reminder there from Al, but nevertheless, the models are tremendous. So we thought in the early days, okay, let's run this competition. We have a big open data set of molecules that of molecules that have been evaluated in blood stage potency assays. We didn't want to prescribe an approach. So we thought, well, just given the actives and inactives, can people derive a predictive model of what we should be making next? So this is back in 2016, 2017. Any approach was allowed. We held back a data set, which was being obtained while the competition was running by Kieran, of a bunch of 400 compounds. And then we knew which ones were active towards the end of the competition, and we asked people to predict which of the compounds, we shared the structures of the pathogen box, which of the compounds should be active, and then judge people on that basis. And 6 competition entries came in, a mixture of public and private sectors. And there were some winners. So a couple of the models managed to predict a few of these molecules. So the actives are shown there on the right, and some of the winners were able to predict 3, maybe 2 of these as being active. But there wasn't a very tight correlation. We think one of the reasons for that is that the original data set did include a bunch of molecules, because it's all of the open source malaria molecules, did include a bunch of molecules which were from other data sets, so from other series that we hadn't, that were not series 4. So people were using models based on biology activity across several series rather than just series 4. But again, we didn't really want to prescribe the way people should be doing this. So this is the result that we got. So we were kind of sitting there thinking, well, it doesn't really, tell us what to make next, unfortunately. And so we sat on this and carried on trying to solve the problem in a sort of more traditional way. But then it came to sort of 2019, and I and others were noticing the extraordinary level of activity that was going into new companies that were trying to apply AI machine learning technologies to drug discovery. So we thought, right, let's do this again. Let's run this project again. We've got a bunch more data now, so the data set's improved in terms of the number of molecules. We have the last entries, which of course are all publicly available, so people have to put their models up online. Let's run this again with the more data, and let's try and get companies involved who have new technologies in this area. And let's get suggestions made for new chemical matter. So not just trying to rationalize known things, but deriving new chemical matter, and we're going to make the molecules, and we're going to test them, and we're going to see how good this is. We're going to see if to be honest, back in 2019, there was a great deal of investment in this area, but there's quite a lot of hype as well. And I was very keen to see whether or not some companies could deliver. So we put it all in the open. We got the funding, which was great, so we could do this. And we again sort of put the competition in the public domain and said, okay, have at it, whatever you want to do. And during the course of the competition, it was very interesting because it's not just about people seeing the competition and then producing solutions. It was about a dialogue that happened during the course of the competition. So that the entrants started talking about it with each other, and in the case on the screen, improving the competition. So our original data set was improved by Willem here and others who saw that actually, hang on, there are some inconsistencies and things that don't make sense here. Let's have a new data set that's got rid of these things and improved it, and people were grateful for that. So there was within competition collaboration, if you see what I mean, and everyone was sharing everything as they were doing it. So it was really fun to see. And in this case, we had 10 models being submitted, again, from a mixture of people, maybe people at universities and the private sector. So you can see that Malomics is here, Mikola's company Oramind, there's Optibrium Intelligence collaboration there, and Excientia were involved. And Vito, who's a long-term collaborator on open source malaria, has a company, Interlink TX, from which he submitted his solution. And the methods were extremely diverse. So there were some QSAR kind of model based on docking to the homology model, which was Ho Leung's approach. But as you can see, there were a bunch of other solutions there, in some cases using proprietary algorithms. And the details are in the paper supporting information. And so what happened is, again, we held back some data. We applied these models to the known data. We determined which models performed the best in terms of predicting activity on the molecules that were held back. And then we asked the winning models, to the submitters of the winning models, to derive new chemical entities that we could then make. And in some cases, the suggestions were quite tricky. So the ones on the top here were suggestions which we don't know if they're any good. We haven't made them because the synthetic effort involved in making these was quite significant. And we didn't have the time or the resources to do that, unfortunately. But there were some other suggestions that came through more based on series 4, which we thought were a bit more tractable within the time that we had. We did ask people to try to involve an increase in solubility aqueous solubility as a sort of ancillary criterion for prediction. But these were some suggestions there, which are more like the sort of core of the series. And we went with these 6 molecules as the ones that we were going to go and make. And so Edwin, my student at the time, made these 6 molecules as quickly as he could and compared it to a positive control that we use in every assay. And you can see that there are some similarities there, but there are some things that we decided were interesting about these molecules, because there were some things that we thought were a bit counterintuitive that we wouldn't have picked. And I'll talk about that in just a second. So we made them all, sent them to Dundee for evaluation of the blood stage potency assay. We also ensured that all the compounds that were submitted hadn't done a scaffold hop. So we had them evaluated again in Kieran Cook's ion regulation assay to check that they were doing the same thing. And the results were this. So there were some molecules that were inactive. 3 were inactive, pretty much inactive. So the Malomix 1 and Davy Guan's compound XC1 were inactive. And 3 were active. So within a threshold that we just installed of 2.5 micromolar as being an indication of activity. And we weren't able to test this Malomix 2 compound in the time we had, but the other 2 were indeed active in Kieran's assay, and the inactive compounds were not. So we're pretty happy with the data. Which constitutes a hit rate of 3 out of 6, which is not bad on a small data set. If you go to the whole of Open Source Malaria Series 4 of what, nearly 400 compounds, then about 165 of those are active at that hit rate, which is a little bit less than the machine rate, but of course it's on a bigger data set. So the performance is pretty good, and we were pretty happy with these results, actually, in part because they were, some of them were quite surprising. So these 2 that I've just circled here, we were pretty sure these would not be active. So, kind of smugly as humans, we sort of said, well, we could have told you that, actually. But it's still worth investigating. One of the compounds here, the XE compounds, we were pretty sure that would be active, but it turned out not to be. So we were a little bit surprised. But this one, the Optiprium one, was really interesting because we would not have made that. We were pretty sure that up in that region where the terbutyl is, we should not put big things there, based on what we knew of the series. So we were very surprised by that compound, and it was really nice to see that was active. Anyway, we think we learned quite a bit here. The result of that is written up now, and we just put it on ChemArchive quite recently, so you can go and see the whole thing if you would like to. Just in terms of the way it works, I guess I often go back to some of these principles of open projects that were made by Eleanor Ostrom, and a similar set of rules actually that came from Benkler in this book, The Penguin and the Leviathan. I'd recommend reading both of these; they're quite interesting. But just in terms of the way the project worked, I think we did align quite well with some of this. In Ostrom's rules, you need ways of monitoring a system if you want a productive collaborative network, and of course we had that because we had the discussion online that was ongoing. And what was very interesting is on the right hand side, this idea of reputation, transparency, and reciprocity is built into one of these open competitions, where people come along with their names and their affiliations and with their data and working methods. So I think it's these describe quite nicely what is it was, I think, a productive way of doing the competition. just in terms of how it works on the human level, you know, again, we had spontaneous contributions, but again, we saw this idea of people collaborating within the competition itself. The private sector guys didn't need to give up their secret source to participate. That was not a requirement to do that, because of course they're proprietary algorithms and they can't compromise those. The private sector nevertheless took part, even though there's a risk to reputation damage by losing. That wasn't really the point here. The point was to see what we could do out of interest to openly determine, you know, capability. It's open, so no one gains a competitive advantage by taking part. It's great to see everyone contributing towards what is a very important problem, you know, development of new medicines for malaria. We saw counterintuitive predictions, which was very interesting. And the co-publication will be a sort of formal reward that's very important to draw a line under the project. And certainly everyone who took part wants to do more of this, which is also really exciting. Because there is a lot more to do. There's constantly screens being done, particularly for tropical diseases, where people identify molecules where we don't know what they're doing, but they have great promise because they kill a parasite or they kill a bug or whatever. And so you're operating in the absence of being able to design molecules rationally. So there's a huge amount we can do in malaria and lots of other things too. And there's lots of other applications for different flavors, I guess, of AI machine learning. For example, in Open Source Malaria Series 3, we don't have a crystal structure of what we think is the target protein, and we need to derive that so that we can sort of work out what the molecule is doing. In the Mycetoma project, which is a fungal infection, we're trying to figure out how molecules can get through a biofilm to be effective in vivo. And the biofilm is extremely complicated molecularly in terms of its structure. And we'd love to know how to get molecules through there, which parameters are important. And one of the SGC projects, Target 2035, which is trying to develop chemical probes for every human protein. There's an interesting project there, which I can tell people about at some other time, where we're trying to, you know, improve a kinase inhibitor so that it can be more selective against one, and trying to target a kinase inhibitor against one kinase whilst worsening the performance against all the other kinases is really hard. But it's an area where I think we need some computational help. So we founded a bunch of projects that are just like this, work on the similar rules. And in many cases, you know, we need these kinds of technologies to help us improve the molecules we're making, particularly when we're working in the absence of a target. I guess the conclusion for me from working on this project in Open Source Malaria is, given that we can predict actives very nicely in the absence of a structure of the biological target, we would be crazy not to be using these technologies in the future. Just in terms of the last few slides, and then I'll finish. Just to make it clear that we are trying to do open source drug discovery all the way through to market, right? So this isn't a sort of hobby project. The idea is to develop a molecule and then take it through to patients, right? That's the idea. It's never been done before. A molecule in the public domain, unpatented, from discovery to market, in the absence of any IP protection. But we're trying to do that in areas perhaps where pharmaceutical industry struggles a little bit to deliver, such as, for example, in the antibiotics field or in neglected tropical diseases in particular. We know that patents aren't necessary to make a big difference to public health. So there are some examples here of molecules that have gone through to market without patent protection, but they just weren't developed in the open. That's all. Itazol is the latest example of a major public health burden that's being addressed by a molecule which is not patented. So there are lots of solutions to this, of how to get a molecule through. What we want to do is try to take molecules that we develop as part of these open projects, and develop them all the way through, and then have some control of the asset so that we could perhaps reimburse or reward people who have contributed, or somehow have a sustainable business model that allows us to make some money back once the molecule, which is unpatented, is on the market. And I and Al Edwards and other people have been really interested in this, about how we can use something called regulatory data exclusivity, which every molecule qualifies for, as a way of protecting a molecule that's on the market from generic competition, and thereby allowing you a bit of breathing room to maybe make some money back, so that you can reassure investors at the start that there is a robust business model. And this is the basis behind this company that you may be interested in, called M4ID Pharma, where we're going to try and work with people to take open assets, unpatented or completely in the open, through to market in a sustainable way. But that's for the future. This is a company that's being founded and is awaiting projects. All right, so just remains for me to thank mostly the people who took part in the competition, which was a really fantastically interesting and fun thing to do. We heard the first talk today, talk about the importance of having fun. This was a real blast. And hopefully the participants found it equally fun. So a massive thanks to them. Massive thanks, of course, to AI3SD for the funding. Something's gone wrong with the slide here, but there's a bunch of other people who've been involved in open source malaria over the years. In particular, Ed here, who did a huge amount on this project and did all the synthesis that I just mentioned. So thanks to them, and thanks very much for your time.